one of you, but if you may start recording, it will be a problem. So I would suggest that one class rep can do so. Okay. In the meantime, let us move on. We have talked about the protein properties, and you have learned what are the primary, secondary, tertiary, and quaternary structure. We will be discussing these structures in further detail. Then we started discussing about the building blocks of the protein, which are amino acids. And you have learned that all the amino acids, they are alpha amino acids, and they exist in L-isomeric form. Those proteins, which is reflected, which is seen, is having several amino acid. Most of the amino acids here, which are originating from the protein, they are going to L-stereoisomer. We have also discussed the concept why D isomers are present in microbial system or in microbial cell wall or peptidoglycan layer, and why it could be beneficial over there. We have also discussed the concept why this D amino acid may be part of the antibiotics. Then we have started discussing about the different kinds of side chains present in the amino acid, and slowly we have learned that the presence of these side chains, whether it is the aromatic amino acid having aliphatic side chain, or the one which has got positive charge, or the one which has got negative charge, all of them, they have certain benefits, have certain role to play. We have also discussed the concept that why, if there are, why in this R group, which you have seen in the previous this one, why there is a different R groups present in the structure. This R group, which is a variable, what they do is that they are involved in the filling the structure when it is in the secondary and tertiary structure form. And what happens actually, suppose I have a very small hydrogen, what will happen when it interacts with another amino acid in the peptide chain? There will be a gap which may be created. Now, to fill the gap, what you will have is that this H can interact with a hydrophobic interaction with a longer chain, say isoleucine, say leucine, and that is the gap which is filled between the protein structure. Then we have talked, okay, so this part I have to start with, and here what you learn is that if I take amino acid and take their spectra, you will find certain extinction coefficient, and that is what is an important one. Have you gone through any thing about the extinction coefficient of a molecule or a solution? Do you have any idea about absorbance?
No, sir.
Okay. I thought this part is being covered in chemistry, but what happens that if you take any solution and pass the monochromatic light through this solution, what happens? Certain amount of the light is being absorbed, and the rest we being transmitted. So what you learn is that each molecule, they have a specific intrinsic property because of which the transmittance occurs, and this is very true. That is why you have different colors. Now, this part you will be doing in lot of experiment where you will be learning the Beer's-Lambert's law, which defines that the transmitted light, the light which is transmitted through a solution, will depend on the property of the solution. This also depends on the concentration of the solution and the path length through which the light passes. So right now I'm not discussing the concept of concentration determination, but what I want to show you here is that the aromatic amino acid, they have a specific absorption spectra, and this spectra reflects that they absorb at 280 nanometers. So here tyrosine and tryptophan, tyrosine is the aromatic amino acid, the one which contains hydroxyl group, and the tryptophan, these two are aromatic amino acid. Of course phenylalanine is also aromatic amino acid. These amino acids, they absorb at 280 nanometer. And that is why if I take a protein solution and I measure its spectra, they will show the absorbance at 280. Similar way, you will find that the nucleic acid also have absorption maxima, or a wavelength at which their absorption occurs. So this just shows you the absorption spectra and gives you some idea that because of the presence of aromatic amino acids, the absorption of protein can occur. So this I am giving you for amino acid. This will be reflected also in protein, because protein will be made up of these tryptophan and tyrosine, at least part, not all, but some of the amino acid will be having tryptophan and tyrosine, and that is why they will show absorption in the range of 280 nanometer. Is this concept clear? Hello, can you
Yes,
hear
sir.
me?
Yes, sir. Yes, sir.
Okay. So what I'm saying is that because tyrosine and tryptophan, which is ingredient of or is a part of your protein structure, since they are absorbing at 280 very specifically, when you take a protein solution, they also absorb at 280 nanometer. Now, this is a list. No need to cram it, but you can see that most of the amino acid they have been given by three-letter code, and that starts with their, so in three-letter code, even if something is starting with A, they can have variable because their three-letter code does not match. So, for example, alanine, we give it ALA, arginine, ARG, asparagine, ASN, because in is there, aspartic acid by ASP. Now, when I come to that single letter code, alanine comes first, so A, it has been assigned A, but others, because they differ in, they are their first, letter is common, and that's why they have been given a different single letter code. This single letter code comes in handy when we have a large number of amino acid which has to be written in a sequence. We will talk about the sequence, etc., at the later stage. Now, coming to the concept of primary structure of protein. Now, what is the primary structure of protein? If you recall, I have shown you certain structure which shows the primary structure of protein. Now, primary structure of protein can be the linear polymer. So first thing is, what are the proteins? If we want to define proteins, proteins are nothing but polypeptides. And this polypeptide, when they're two amino acid, they will join by a peptide bond. So it becomes dipeptide. When you have several amino acid linked together, they make polypeptide. And when this polypeptide has a number 50 and above, we call them as a proteins. So let us see how this happens. So proteins are linear polymers which are formed by linking of alpha carboxylic group of one amino acid to alpha amino group of another amino acid with a peptide bond. So suppose we have one of the amino acid. This is amino acid number 1. This is amino acid number 2. So how the interaction will occur? The carboxylic group of the first amino acid, this is the carboxylic group of the first amino acid, that will interact with the alpha amino group of another amino acid. So alpha carboxylic group of, so carboxylic group interacts with the amino group, they form a bond, and this bond is CONH bond or peptide bond. What do you call in chemistry? What is this bond called as when you have a carboxylic group interacting with amino group?
Amide.
Yeah, we call it the amide bond. Here we have referred it as a peptide bond. So it's a nothing, different. You have already learned about it. So what happens that here, when these two interact, one molecule of water is being removed. And so one OH from carboxylic group and one hydrogen from NH2 group, if they are removed, they form peptide bond, and that is CONH bond. So in a protein, what do you expect? Or if there are many amino acids, they are linked together, what do you expect? You expect that these peptide bonds will be several. So look at this structure. You have one peptide bond, two peptide bond, three peptide bond. So the number of amino acid will decide how many peptide bonds are going to be present. So a series of amino acids, when they're joined by peptide bonds, we will call them as a peptide chain. So this chain which you are looking at, this chain is a peptide chain. And that's why we can call it a polypeptide. If one peptide bond is there, you have, say, two amino acids joined together, you can call it a dipeptide, you can call them a tripeptide if there are three. And if there are many, we call them as a polypeptide. Now, each amino acid here, suppose I have a polypeptide chain and you have this amino acid. What is this amino acid? It is written below. It is a tyrosine. It's a phenylalanine having OH group. So it is a tyrosine. So the amino acid link here is not called as amino acid. They will be called as a residue. Why residue? Because it is not a complete amino acid. It is amino acid minus part of the water molecule. So that is why each amino acids are referred as a residue in a polypeptide chain. That is true with other polymers as well. Uh, by convention, and this is just convention, that when we write the chain of amino acid, what we do is that we put amino end. The amino end is taken in the beginning of the polypeptide chain. So here, what you see is that this amino group, this group, is in the start and the carboxylic group is at the end. So what we call them? We call this amino terminal or N terminal. N is for because of the N NH2 group. So we call as N terminal and this will be called as a C terminal. So each peptide chain will have N terminal and C terminal, and because of the convention, what we call them, the way we write them is that we start with the N terminal, end with the C terminal. Okay. Now, this is what has been shown in this picture. This is another polypeptide chain. You will be learning that, this group is on one side, this group is another side. So we will talk about what is the trans orientation and what could be the cis orientation. These things we will take up a little later. Now, sequence of amino acid in a polypeptide chain, as you have learned, starts with amino terminal, but this is not a necessary event. You can write the way you want, but as a convention, we always write N terminal first. Now, coming to the primary structure, you have learned that the amino acid, they are linked together by peptide bond, and that's why they are referred as the primary structure. Another very important thing comes in the primary structure is the other type of covalent bond present in a peptide chain. So what could be the other type of other type of covalent bond present in the peptide chain? It could be, these bonds could be present because of presence of a sulfhydryl group. Yesterday we have talked about whenever sulfhydryl group is present, which is present in one amino acid called as a cysteine, and these are very reactive groups. So what happens that this group has a tendency to react with the other similar group present in the chain. So if SH group is present here, SH group is present here, they will react. Now, this reaction leads to the covalent bonding. So see, you have a covalent bonding. Now you may have a question whether this cysteine uh, can interact within the same molecule or different molecules. Suppose I have a single chain, and if there are two cysteine, will they react? Answer is yes. If there are two different chains and cysteines are present there, if they come in vicinity, they will react. So actually, cysteine being very reactive, SH group being very reactive, can interact with another cysteine residue and they form cysteine. So this amino acid, if it is isolated, of course, they can have SS bond together. They can be called as a cysteine. In most of the natural polypeptide chain, what you find is that the polypeptide chain contains something like 50 to 200,000 amino acids. If they are less than 50 amino acid, by convention, they are called as the peptides or polypeptides. Then if you have amino acid more than 50 and around most of them, they will be within 2000 amino acid, they will be called as a protein. So proteins are nothing but polypeptide above certain number, though this number has been given for, some understanding, but there is always, polypeptides, they are considered as a proteins. The mean molecular mass, if you look at the molecular mass of individual amino acid, it is around 110. And because there are 50 to 200, the most of the protein, they will be having molecular weight of 5500 to 22,000, 220,000. Now, these linear peptide chain is a cross-link. So when I talk about the cysteine-cysteine interaction, that is mostly for the cross-linking within the peptide chain or between the peptide chain. So now this figure can give you the bottom one, which is the structure of the insulin, bovine insulin. It gives you the idea how the interaction could happen. So you see a residue, which is as a sixth residue and 11th residue. Both of them, they have their, uh, the bond, which is these bonds are between, this is the S of sixth residue, this is the SH group 11th residue, they can interact with each other and they can form disulfide bond. On the other hand, you can see these residues, they can interact with the intermolecular chain. So there is another chain within the same peptide, they can bind together. So what you have learned is that the primary structure not only involves the peptide bond, they also include the disulfide bond. In other words, the primary structure refers to the covalent bonding present in the protein chain. Now, coming to some more interesting points about the proteins, you have each protein which has unique and precisely defined amino acid sequence. Now, question comes, what are the amino acid sequence? Do you understand amino acid sequence? Have you any idea about amino acid sequence? See, sequence of amino acid, if I take this chain, the chain which I'm referring to, this chain, and break all the peptide bonds as well as the disulfide bond. What we get is we get certain number of glycine, GLY refers to glycine, ILE is isoleucine, and that way you can name all the amino acids. So in this chain, if I break all the peptide bonds and disulfide bond, you get a set of amino acids. These amino acids, when we analyze, you get, say there are acid, 2, 3 leucine or so. I'm just putting a random number. So this number is being generated. So by analyzing, what you find is that there is a certain number of cysteine, certain number of serine, certain number of glutamine, and that's the presence of amino acid in a particular protein or a particular peptide chain. Does it mean a sequence? Answer is no. This refers to the composition of amino acid. When I refer to sequence, I specifically means that there is a sequence where first is a glycine, followed by isoleucine, then valine, then glutamic acid, then uh, glutamine, then cysteine, and so on. So, sequence of amino acid precisely defines the arrangement of amino acid in a unique sequence. And this is always unique. That means you may have two proteins which may have the same composition but may differ in their amino acid sequence. Am I clear to this?
Yes, sir.
Okay. Now, how the sequence is determined, how it happens that there are 20 amino acid and you get a very specific sequence. What makes them to be aligned in a particular fashion? Answer is the sequence of nucleotide in DNA So DNA is the one where there is a sequence of nucleotide, which we will discuss in when we talk about the DNA structure. That is a specific. It determines the molecular heredity, how the things should be inherited from one generation to another generation. So what happens? DNA forms a complementary sequence of RNA, and these RNA define specific sequence of amino acid in a protein. In particular, each of the 20 amino acids of the repertoire is encoded by one or more specific sequence of three nucleotides. This is a slight tricky. When I talk about the DNA, when we talk about the nucleotide, you will understand that in DNA, suppose I say DNA is made up of nucleotides. So in a chain now, so DNA is nothing but is a polynucleotide. So now in this chain, if I say three sequence of a three of nucleotides, they define which amino acid they should interact or which amino acid they will be binding. So in RNA sequence, not in the DNA, DNA makes a complementary RNA, and the RNA will have three codes which can bind with a particular amino acid. I will talk about this when we are talking about the DNA and their function. But here one should understand that if glycine is binding, suppose this is a glycine, it will have three nucleotides. Here you have one, two, and three nucleotides which are specific for binding to the glycine. So I'll tell you how this binding occurs, how the specificity occurs. But that's the way it happens. And so nucleotide sequence can determine the sequence of amino acid. So that's why you have very precise sequence of amino acid in each protein. Now, knowing the amino acid sequence, why should you learn? The question comes, why should one learn about the amino acid sequence? What is the importance of this? Answer is that by understanding the sequence of a protein, it usually gives a mechanism of action. Why? Suppose I have a catalyst, which is a biological catalyst or enzyme. Now, if I say that these three residues are important for interaction or binding with the substrate, how can you define unless you show that these amino acids are involved in the binding? So that's why the amino acid sequence, especially the sequence at the catalytic site, becomes important to determine their mechanism of action. The amino acid sequence also determine the three-dimensional structure of the protein. This part you will learn in the next few lectures, where the sequence of amino acid determines the tertiary structure of the protein. Tertiary structure means the folded structure. I will define it further, but three-dimensional structure or tertiary structure of the proteins are being determined by the primary structure of the amino acid. So it is very important that we must understand what is the primary structure of protein. Then in some cases, you see that the sequence also determines the molecular pathology. What is molecular pathology? If I say that if I replace glutamic acid by some other amino acid, it becomes it becomes a mutation in the gene, because of the mutation of gene, and that can cause a particular disease. So that is called as a molecular pathology. This is well reflected in case of sickle cell anemia. Now let us move on to how these polypeptide chains are formed and what are the flexibility, why should they move. So answer here is that the peptide bonds are essentially planar, and the peptide bond has a considerable double bond characteristic. Why should we call them that peptide bonds are having double bond characteristic? In the previous slide, what we have shown is that these are the peptide bonds. So this is the peptide bond. I will be, discussing this further, but these are the peptide bonds. So this is the peptide bond we are referring to. So this is the peptide bond. Now what I'm saying is that these peptide bonds, CN bond, the bond between CO and NH, I'm not talking about NH and C. I'm talking about this bond which is between CO and NH. So this is the peptide bond. This peptide bond has a relatively a double bond, the characteristic of double bond. Why it is so? because the distance in a peptide bond is typically 1.32 angstrom, which is very similar to, so this is the typical distance of your peptide bond, which is in between, which has a characteristic between C and single bond, which is 1.49, and C and double bond. So it is smaller than the single bond, but larger than the double bond, and that is why they have characteristic similar to the double bond between C and N. So because of this, you will not have CN bond rotating. So what are rotating? So that part we'll discuss. There are two configurations which are possible for planar peptide bond. That is, one is a trans configuration and another is a cis configuration. So we'll discuss these two configuration in the peptide bond. And this kind of restriction is because of the steric hindrance, and that is what we'll talk about. So almost all peptide bonds in protein, they are in the trans conformation, or their configuration is a trans form. And this preference of trans over cis can be explained by the fact that the steric clashes between the group attached to alpha carbon atom hinders the formation of cis form, but do not occur in the trans forms. In the next few slides, I will try to explain. So here you have this is CO bond, there is NH bond, so there is a peptide bond. Now this peptide bond is having double bond characteristics, so their movement is less. You can see it here. It is the one which is having lesser movement. So this bond is relatively less flexible. But the other bonds I'll just show you in the next slide, which are much more flexible. So what are the bonds? So this peptide bond is, sorry. Okay, so so this is the peptide bond, which is restricted bond, whereas the bonds which are, for example, this bond, and this bond could be flexible. So what you have is that the bond which is between CO, so this is CO and NH, this is the fixed one, and the one between N and CH, this one, and the one between CC, this one, they are flexible. These bonds are referred as a phi bond. So look at this bond between NH and CH, and another between CO and CH. So these two bonds, they are flexible and they are referred as a phi or psi bond. Now this phi and psi bond, they have a rotational angle. So they have a possibility of different types of conformation, and they will interact with the main chain of the protein. So if I say the protein, this is the main chain of the protein, right? And the one which is R group, they are making these side chains. So what you will find is that these side chain, they interact with the main chain or the other, residue present in the main chain, and that is why the folding occurs. Now, how these can fold? They can fold with a certain angle, and these are given by phi or psi bonds. Now, there are certain rotation which is given. It is both depending on the parallel or anti-parallel. So you can have flexibility of minus 120 to plus 120, and that is the way it is being defined. Just a second. So what you have is you have a flexibility of the structure from minus 180 plus 180. I'm sorry. So this is what we were discussing. Now in this structure, what you see is that this phi bond and this is the psi bond, which are, rotating. And as we discussed in the previous, discussion, that there are, okay, let me just pick up the pen. So this two group, this is around CHR, and this is also CHR. Now, if they are two away from each other, facing away from each other, that is a trans form. And if they are towards each other, they are cis form. So in cis form, what happens? They are very close to each other and create a steric hindrance. And that is why in most of the protein, it is in the trans configuration. So this is the reason for trans configuration. Now let us, so what you always see is that though there is a possibility that the orientation could be anything, that it could be cis or trans, but always you see that in protein, they are mostly in the trans form, right? Now coming to the next slide, here we will define it further. The freedom of rotation about two bonds of each amino acid, they allow proteins to fold in many different ways. So because of the phi and psi bond rotation, they can be of different types of interaction you can see in protein structure. The two rate rotation about these bonds can be specified by dihedral angle. This dihedral angle is nothing but the one we have talked about, phi bond and psi bond. The angle of rotation about the bond between nitrogen and carbon, alpha carbon, this A is refers to alpha carbon atom, is called as a phi. And the one between alpha carbon atom and carbonyl atom is called as a psi. This is what we have discussed. So between the this one between the carbonyl group and N is called as a phi, and between the carbonyl group, so between the carbonyl group and the carbon bond is known as psi. So these angles, they are rotating, and that's why you have different types of folding pattern in the protein. Okay, before we discuss this, let us further discuss about the phi and psi angle. A clockwise rotation about either bond is viewed from the front and the back group corresponding to the positive value. That is why you have seen a positive value and then you have a negative value. The phi and psi angle, they determine the path of the polypeptide chain, the way the polypeptide chain interact with each other, the way they can fold. This fact has been, discovered by one of the renowned scientists from India, who is G.N. Ramachandran, and he has given the characteristics. So what he did is that he has tried to plot between phi and psi angle, and he has given which one could be the possible or which could be the most favorable secondary structure of the protein. So the allowed value can be visualized in two-dimensional plot, which is called as a Ramachandran plot or Ramachandran diagram. The three quarters of possible phi and psi combination are excluded simply because of the local steric classes. So basically this plot, GN Ramachandran plot, it gives you steric exclusion. That means which kind of interaction, which kind of a folding is not possible. He has given a very important fact, that the fact that two atoms cannot be in the same place at the same time. And this could be a very important tool to predict the secondary structure. So here, this you have already learned about the phi angle and psi angle. Now let us see Ramachandran's plot. So Ramachandran has given plot for alpha helix, beta sheet, what are the types of possibility in the interaction. So you can see that the dark green part is the most favorable conformation, whereas this other part where it is a light one, it is a slightly lesser possible conformation. And then the one which is red, it is excluded or very rare, and the other one is not possible. So this gives us a way to exclude the different type of interaction. So what he has done is that he has taken psi angle and phi angle. He has plotted between the psi angle and phi angle, and this plot shows that what are the possible secondary structure or conformation is theoretically can be predicted, or theoretically you can say that angle rotation could be up to this one possible, otherwise there will be steric hindrance. Now with this I have covered the primary structure of the protein, the possible rotation in the primary structure of the protein. So what is the learning, what you have learned today? Today you have learned about the formation of the peptide bond. You have learned that how the protein sequence is written, what is the primary structure of the protein, how this primary structure can fold and ultimately give rise to the secondary structure. We have not covered the secondary structure. In the next lecture, we will talk about the secondary and tertiary structure. But here you have learned that why the proteins they form the secondary structure. What leads them to interact within the chain. So you will be learning that the proteins, if it is a polypeptide chain, it never remains as a straight chain. It always folds back. So naturally, the proteins they are not existing as a just straight chain of a polypeptide. They are a folded structure. And the complexity in the folding occurs because of the R chain. So you have a chain of a polypeptide where you have a CONH bond, and this CONH bond is a repeated. That means you have a repeated peptide bonds. So what will happen that there will be certain interaction which will be neighboring interaction. That means amino acid which is present in the peptide chain, their CO group can interact with the NH group. How do this CO and NH can interact in a protein structure? What type of bond they will form? Suppose one of the amino acid here, let us look at one of the amino acid. Okay, so let us see this structure. I have this amino acid, number one, there is another amino acid. How these amino acid can interact with each other? Can they interact?
So hydrogen bonds.
Yeah, they will have a hydrogen bond, and the hydrogen bond will be between the carboxylic group and the NH group. So NH will be hydrogen donor and CO will be hydrogen acceptor. But question is that every CO and NH will interact, or there will be certain distancing? So this part we will discuss. Approximately 3.6 residues apart from each other, these amino acids they fold, they make a helical structure, and they start interacting with each other, and that's why you get helical conformation of the protein. So when we talk about the secondary structure, you will be learning that within the chain there can be interaction. Now there can be hydrogen bonding, which can also lead to the secondary structure, which is not alpha helical structure, could be a instead of helix, it could be a pleated structure. So we'll talk about what are the beta pleated structure. So we'll talk about different conformation which is arising because of the primary structure. So now question comes that if I say that these are the repeated sequence, because of that there is a such kind of. Now, in addition to that, there will be different types of R groups. So you have different R groups which are present in the, suppose in this case, let me just take an example. So this is one R group, which is from tyrosine. Then you have another R group, which is a glycine. Then this is again glycine, another R group. So these are R groups. When these R groups interact with each other, then what you will have is you will have, so these are R groups. Now the rest chain, which is without R, they are making the main chain. And these R group are side chain. So it is the side chain which interact with each other and gives the different structure and different folding of the protein. So I think this concept should be clear to you that the secondary and tertiary structure are determined because whatever the primary structure sequence we have, they will have a specific R group, and this specific R group can come in contact with the specific R group within the chain, and that can lead to the folding of the protein. So the folding of a protein is based on their primary structure to a major extent. But you cannot predict the structure to 100%. The reason is that this interaction is a flexible and can vary, and that's why we cannot 100% we cannot predict the structure. So with this, I'm ending my class. I will be signing off. And if you have any question, you can raise. We have 1 or 2 minutes. Otherwise, you can raise the question in the next class. Any question? Okay, if there are no more questions, I will be signing off. Okay, bye. So who was recording? Let me know who is recording, who is keeping recording. Hello?
Hello, sir.
Yeah, who is recording?
Sir, myself Narendra Kumar.
Okay, Naren. So what you will do is that you can share it. Either you can share it with me and I can edit and give it to you, or alternatively, if you want, you can share, keep it and share it with your friends, right?
Okay.
So I'm stopping at this point.
So I had one more doubt.
Yeah.
So in the beginning, you said that you'll be giving us some assignments. So when will you start giving us the assignments?
This assignment, I'll let you know. It will
Okay.
be in the form of assignment. It could be form of a quiz. So that's the way we will start with.
Okay.
So the quizzes will be announced, right? Or will they be
Yeah,
surprise
yeah.
quizzes?
It will be announced. But one very saddening part is that the number of people joining the class is very less. And that is what prompts me that I should have quiz in the unannounced class. So it is important that all of you, unless there is a very specific reason, all of you should join the class. Because I am giving you all the PowerPoint presentation that is already available to you. Now, with this, I think you should not get the flexibility, because this time you have a flexibility. It does not mean that you do not attend the classes. And as such, even if you record, even if you take the PowerPoint, you will not understand the concept unless it is explained to you. So I would request all of you to attend classes. Okay, now I have another class, so I'll be signing off. Okay, bye.
Thank you, sir.